So back in the 80s, there was a big outbreak of HIV in patients of a Florida dentist. And when the dentist passed away, it turned out that he tested HIV positive. And so there was this big sort of controversy trying to figure out whether or not the patients had gotten HIV or contracted HIV from this dentist, or whether they had gotten it somewhere else from the community, and this was just a coincidence that all this cluster of HIV positive individuals was associated with this dentist. But this was a very difficult question to answer until some scientists came up with the great idea of using phylogenetic trees to address this question. And so what they decided to do was basically from each infected patient, they took 2 HIV samples. So they got 2 samples of blood, and they genotyped the HIV in that sample of blood. Then they got 2 samples from the dentist and looked at the DNA sequences of his strain of HIV. They also got HIV samples from local HIV positive individuals that were not patients of the dentist, and then finally obtained a less related strain of HIV from Africa. And they compared the genetic distances of those samples. When I talk about genetic distances, what I'm talking about are those matrices that we came up with in the last lecture, where you're looking at how closely or how similar are the DNA sequences of those different strains. And then they build phylogenetic trees based on those genetic distances. Okay, so given that, if the patients contracted HIV from the dentist, which of the following would you predict to see in terms of what the phylogenetic tree should look like based on the genetic differences of those different strains of HIV? So the correct answer to this one is in fact C. So what we predict is that the patient strains and the dentist strains should be most closely related to each other, more so than they are to either the local or the African strain. So we would predict this, that the dentist and the patient strains look the most similar in terms of their genetic sequences and have the smallest genetic distance. And if the patients did not contract HIV from the dentist, which of the following would we predict to see? We would expect to see D. And so here we see that the dentist strains are not as closely related to the patient strains as local non-patient HIV strains are. So in this case, this indicates that they likely got those strains through contacts outside of the dentist. So just from the community as opposed to from the dentist. So this is what they actually found. So here you can see dentist strain 1 and 2 and different patient strains. And then here you can see different local strains and patient strains, and then the outgroup of the African strain of HIV. And so you can see strains of HIV from patients A, B, E, C, and G are more closely related to the dentist strains than they are to these local strains here. So they have a more recent common ancestor and are therefore more closely related to the dentist strains than they are to any of these local strains. Whereas strains from patients D and F are more closely related to these local strains. Okay, so which of the following do these results support? And C is the correct answer. So it looks like patients A, B, E, and C, and G probably did get their infections from the dentist because their strain is more closely related to the dentist strain than it is to local strains. Whereas patients D and F, it looks like there's less evidence to support that. As a result of this, there was some heavy legislation put through where congressmen were basically trying to pass legislation restricting the activities of persons infected with HIV, and also mandating that some people involved in healthcare were required to take HIV tests. And some of that legislation was pushed through, some legislation was not, and you can imagine the sort of ethical issues that are associated with all of that. And so there were a lot of sort of social and ethical implications that resulted from this case. And so this is just one example of how creating phylogenetic trees and understanding monophyletic groups and the importance of creating these sort of supported monophyletic groups can be vital for really sort of practical matters. And we see that not only in that example of these HIV tests, but also in the DNA barcoding stuff that you guys are doing. So this is a great example of how combining these sort of phylogenetic trees with taxonomic identities can be of practical use. And so these are actually data from a Bio 150 student project where they looked at the identity of red snapper. So they went and bought red snapper at a couple local food stores, and they basically said, what is the DNA barcode identity or the phylogenetic tree of the DNA barcode identity of these snapper samples tell us that the actual taxonomic identity of those samples is. And what they found was a couple different clades popped out. And so here on this tree over here, you can see this is a student snapper, red snapper sample. And these are 1, 2, 3 other red snapper samples. And we've got one clade that pops out here that is in fact associated with other snapper species. And so in this case, they were able to say this is actually red snapper based on the genetic similarity that this phylogenetic tree says this sample is more closely related to snapper species than it is to other species. The other samples that they had, however, came out in a clade or a monophyletic group associated with, these are all species of tilapia, which is a much cheaper fish. So even though it was sold as red snapper, it actually turned out to be a different fish species. And so here you have an example of combining taxonomic knowledge with these phylogenetic trees to get the actual identity of different fish samples that have been collected around town. And this is a trend that's seen not only here in Harrisonburg, but sort of all over the place. So this is from a larger study based on LA sushi restaurants looking at the percent of fish mislabeled from 2012 to 2015. And they found that 47% of fish had been mislabeled overall, with trends across different species, especially with red snapper and halibut and yellowtail and yellowfin tuna. So this is a trend that we're seeing not just locally, but all over the place. These sorts of phylogenetic analyses are also being currently used for COVID-19 analyses. And so this is looking at different COVID strains that were collected from Washington that you see in pink, California that you see in blue, China that you see in green, and New York that you see in yellow. And basically what they did is looked at the genetic similarities of different strains of COVID in these different geographic locations. And what they found is that 85% of the viruses collected from Washington state were genetically similar and popping out in this individual clade that's within these sort of Chinese strains. And so they took this to be evidence that there may have been a single or one or few individuals that came into Washington state with a Chinese strain, and that strain from there spread like crazy within Washington state. And so there you had an actual Chinese strain that could maybe potentially be traced back to one or a few individuals. Whereas when you looked at the other strains, so we have some other Washington strains here, some New York strains here, California strains here that are sort of popping off in this other group. And they're saying that that's evidence that perhaps these strains are from European introductions or multiple different introductions of a different strain based on them breaking out in this different group here. And so they're using that to sort of backtrack and analyze potentially where different strains of the virus of COVID-19 are being introduced from.